tasks where you have A and B. So don't guess B, no penalties for guessing. Survey question that we do every couple of lectures is about the pace. So again, this does not change the depth or amount of material no matter what. It's still the same lecture, still the same notes, still the same textbook readings and exam. That being said, again, I can go as fast or as quick as you want. I can finish the lecture in 10 minutes and I'll go home and watch TV. I can take 4 hours. I don't really care. It's really what works best for you. So that being said, we do have the third hour to work with. So let me read through the options. And we do this every couple of lectures, right? We already did it once. So A, the lecture pace is too slow. The people who need extra help can stay for the third hour, read the textbook posted on Blackboard. B, lecture pace is about right, stay about the same. If I need help, I'll stay for the third hour. And then C, the lecture pace is too fast, let's go slower. So it really doesn't matter to me, it's really just what works best for the majority. So A, who says the lecture pace is too slow, let's go a little bit faster. B, about the same, stay about the same. Okay, and C, it's a little bit fast, it's a little bit slower. So I kind of have to go with the majority. I know there's a couple of people here and there who want it slower, faster, but I just have to really go with the majority. Okay, so this class is all about protein sorting. We'll talk about protein sorting for the nucleus, mitochondria, and chloroplast, peroxisome, for your readings. We have a bit of a long class today, so I'm going to try to write as quickly as possible, but we'll go through it a step at a time. There's no preamble, just because it is a little bit long. So this class, we're going to talk about protein sorting. Again, you can't change your DNA sequence, you can change what comes out of your DNA. We'll talk about protein sorting. Now, the first thing we want to review are the typical parts of the animal cell. Now, you should know these variety of different parts. We're not going to go through it now. If you don't know, you can use Google or use Wikipedia. A couple of things we need to talk about. The animal-specific parts. So first, you have the lysosome. So degradation of cellular parts no longer needed. Lysosome functions in the degradation of cellular parts no longer needed. Lysosome, degradation of cellular parts no longer needed. Now you also have the extracellular matrix. The extracellular matrix is basically, it's the stuff outside of cells. Now if you look at most, not all, if you look at a lot of multicellular organisms such as yourself, such as plants, you're more than just a sack of cells, right? You have stuff outside the cells as well. Well, for animals, the stuff outside the cells is a very specific composition, and we call that extracellular matrix. Now, in a later class, we'll go over the details of what is in this extracellular matrix. But just note now, it's a very specialized stuff outside the cells in animals. Plants will have stuff outside their cells too, but it's a very specialized composition in animals, extracellular matrix. On the next slide, you have the typical plant cell. So obviously, on the column over here, this is going to be common to both animals and plants. Right? No one in this class should look at a nucleus and go, oh, that's only in plants, right? No, right? That's going to be in both animals and plants. ER, Golgi, peroxisome, mitochondria, plasma membrane in both plants and animals. Now, you have a couple of things that are plant-specific. Be careful here. This is plant-specific compared to animal cells. So write in compared to animal cells. Plant-specific compared to animal cells. You'll see what I mean in a sec, but this is plant-specific compared to animal cells. What do I mean by plant-specific compared to animal cells? Well, cell wall, that's going to be in plants, not in animals, but other species, other organisms can have cell walls, right? Bacteria can have cell walls. So this is not like it's only in plants. Plant-specific relative to animals. So cell walls, plants, yes, animals, no. Other organisms can have cell walls. Now, cell wall, you can write down tough protective outer coat. Should be quite obvious. Tough protective outer coat. You have a vacuole. There are two types of vacuoles. The first type functions for storage. Again, first type for storage. Second type, like the animal lysosome, for degradation. Second type, it's like the animal lysosome for degradation. Chloroplast for photosynthesis. Right, I always say, if you don't know what photosynthesis is, just find some fitting grade 4, you need to ask me. Okay, so again, cell wall, tough protective outer coat, vacuole, two types, one for storage, second type for degradation, like the animal lysosome, and chloroplast for photosynthesis. Couple of extra terms that you make sure you distinguish between cytoplasm, cytosol, and lumen. They're not the same thing. Cytoplasm and cytosol is a little bit confusing. You can kind of confuse them, but lumen is going to be a little bit. Now, looks like most of you are done talking. So cytoplasm and cytosol, they're not the same thing, although you'll often hear them used interchangeably. You'll hear professors like myself, like the morning lecture, kind of mix them up a little bit, but they're not quite the same thing. So cytoplasm is the space outside of the nucleus. It actually includes the organelles, with the exception of the nucleus. Right? It includes the organelles, but of course not the nucleus. Cytoplasm, again, includes the organelles, but really not the nucleus. The cytosol will include stuff like ribosomes, will include the cytoskeleton, but does not include membrane-bound organelles. Again, cytosol includes stuff like ribosomes, includes the cytoskeleton, but does not include membrane-bound organelles. All right, so again, cytoplasm space outside of the nucleus, really you're including all the organelles here with, of course, the exception of the nucleus. And then cytosol, it's a little bit less. You include ribosomes, you include the cytoskeleton, but you don't really include the membrane-bound organelles. So cytoplasm really is a bit more. Lumen is the aqueous part inside organelles, so like inside the endoplasmic reticulum, that would be the lumen. Now this class really focuses on protein sorting. You're going to move proteins within the cell from different compartments, out of the cell, and then into the cell. So again, protein sorting, so obviously you're moving proteins around. You're going to move them to different compartments. You can move them out of the cell, you can move them into the cell. Protein synthesis starts on ribosomes in the cytosol. That should not be a shock. Proteins must be sorted to the correct location. Here is your cell, all sorts of different cellular parts, cellular organelles. Here is the cytosol. That's where you start your protein synthesis, but you'll move proteins to different locations. Protein sorting can be post-translational and co-translational. Post-translational means what the name really says, right? After translation. Proteins have to be fully synthesized in the cytosol before sorting. Co-translational, that really refers to what's happening with the endoplasmic reticulum. Protein sorting, again, can be post-translational. So post-translational means after you translate it, then you sort it. Or co-translation, as in while you are synthesizing the protein, while you're translating, you're sorting it. Be careful, within post-translational sorting, there's two types, unfolded or folded. Unfolded is for sorting to mitochondria and plastids. Folded is for nucleus and peroxisomes. So let's take a look at one example protein shown for you right here. This protein is being sorted to the nucleus. So if you see a protein being sorted to the nucleus, you know it's post-translational. This protein is fully translated before it's sorted. As well, the protein is properly folded before it's sorted. So this protein see it actually exposes what's called a nuclear localization signal. Then you move this thing to a nucleus. That's very different from co-translational process. Here you are making the protein as you are sorting it. That happens for the endoplasmic reticulum. You can see here is a ribosome translating the mRNA into a protein, and while you are translating the protein, you are moving it into the endoplasmic reticulum. The overview for sorting is shown for you here. This is the same slide that you've seen in Bio 130 if you took the prerequisite course. Two things to note here, the colors and the arrows. So if you printed it off in black and white, take a look at the colors, it's color coordinated. Take a look at the arrows too. Red is gated transport. Notice gated is between the cytosol and the nucleus. It can go both directions. You can go into the nucleus, you can go into the cytosol. Both directions for gated. Transmembrane in blue. Here you require protein translocators. It's only one way. Cytosol to the mitochondria, ER, plastids, or peroxisomes. You don't really go back. It's a one-way trip. Transmembrane transport. Vesicular transport is in green, where vesicles move protein between compartments. Notice they're not all going both directions. Most of them are going both directions. You can go back and forth from the ER to the Golgi, back and forth to secretory vesicles, but some of these are one way, like secretory vesicles to the outside of the cell. You don't really go back into secretory vesicles. Okay, so again, take a look at your overview for protein sorting. Look at the color. Some are gated, some are transmembrane, some are vesicular. Look at the direction of the arrows. Transmembrane is all one way. Gated can go both ways. Vesicular really depends. Largely both ways, but not necessarily always both ways. So we're going to focus first on gated. We're going to go through the different ones. Gated transport, again, between the cytosol and the nucleus. You can go both directions. You go through nuclear pore complexes. They'll selectively transport macromolecules, where smaller molecules can freely diffuse through. For macromolecules, you can think of stuff like RNA, protein. So for nuclear pore complexes, again, larger macromolecules, you get selective transport, RNA and proteins. That's what you think of as large macromolecules, just macromolecules. Smaller molecules, stuff smaller than RNA, smaller than proteins, can freely diffuse, less than 5,000 Kelvin. To travel through this, you have to travel through this nuclear pore complex. And again, proteins can go in both directions. You get nuclear import, which goes from the cytosol to the nucleus, and nuclear export from the nucleus to the cytosol. So this is not like rocket science. I think it's pretty simple, right? Nuclear import, as in going from the cytosol to the nucleus, going inside the nucleus, and then nuclear export, leaving the nucleus, going to the cytosol. Here's your standard nuclear envelope. You can see this side is the cytosol, this side is the nucleus. Now the nuclear envelope, don't forget, is actually two membranes. So you have an inner nuclear membrane. This thing is a phospholipid bilayer. And you also have an outer nuclear membrane, another phospholipid bilayer. So it's in fact two phospholipid bilayers. One full inner nuclear membrane, one full outer nuclear membrane. Here is the nuclear pore complex, NPC for short. There's a lot of proteins that make this up. About 30 proteins make this up. We're not going to go through the different proteins that make up this nuclear pore complex. If you're so interested, you can read about it in your textbook. You basically have these fibrils that help attach to a variety of different proteins. And as well, in here, you have nucleoporins, a type of protein. You'll see that again, nucleoporins. So I just want to give you an idea of how impressive this transport is. Like, if you just kind of look at the nuclear pore complex, you're like, okay, that's nice, right? You move stuff in and out of the nucleus. But in fact, it is extremely impressive. You can read about this in your textbook. Just one of these can transport 1,000 macromolecules per second in both directions. You can imagine 1,000 different RNA and proteins going in both directions per second, and there's no crashes, which is, again, very impressive. Like, if you imagine a highway, 1,000 cars per second, there's going to be a crash, right? Going in both directions. Really doesn't happen in a nuclear pore complex. It's even more impressive one nucleus having 30 to 40 different nuclear pore complexes. So, or sorry, 3,000 actually, 3,000 to 4,000 different nuclear pore complexes. So each one of these transporting 1,000 macromolecules per second. It's really a lot of transport between the nucleus and the cytosol. Now the first thing we want to talk about is nuclear import. This is moving stuff into the nucleus. And I just want to say a quick word here, because what confuses a lot of students is the term cargo proteins. Cargo is just like the lay definition of cargo. Like if you move stuff on a bus, if you move stuff on a train, move stuff on an airplane, it's cargo, right? Think luggage. So same thing for proteins. Cargo proteins are just proteins that go somewhere. Cargo proteins can go to the nucleus. Cargo proteins can go to the cytosol. Cargo proteins can go to mitochondria. Doesn't matter. The protein that you're moving, that's cargo, right? That's just a lay definition. It's not like a real scientific strict term. So cargo, the protein that's moving. Now, if we're talking about nuclear import, as in going into the nucleus, yeah, cargo proteins will go into the nucleus. But cargo proteins in general don't have to just go to the nucleus, right? As long as the protein moves somewhere, it's a cargo protein. When we talk about nuclear import, yes, the cargo proteins will move into the nucleus. So I think it's just a bit of an English definition. Cargo, think luggage, it moves somewhere. Now, nuclear import, moving into the nucleus, that requires proteins called nuclear import receptors. If you are moving into the nucleus, if it's nuclear import, then the cargo protein, the protein that moves, has what we call a nuclear localization signal. It's a signal that says, take me into the nucleus. Tells the cell, take me into the nucleus. The nuclear import receptor will bind to that signal. It will bind to the nuclear localization signal. And then it will also bind to nucleoporins, proteins in the nuclear pore complex. Okay, so I know for like 75% of you, you're all with me, because no problem with cargo proteins. I'll just repeat it one more time, just because it gets a lot more complicated. I don't want to leave like 30% of the class behind. Nuclear import, you're moving into the nucleus. Cargo proteins, proteins that go somewhere. In this case, if we're talking about nuclear import, cargo proteins are going into the nucleus. If you're moving into the nucleus, you have to have a nuclear localization signal, an NLS, a signal that says, take me to the nucleus. The nuclear import receptor will bind to the nuclear localization signal. The NLSs, they're rich in lysine and arginine. So the signal that says, take me to the nucleus, they're rich in lysine and arginine. Now be careful, it's not any sequence that has lysine and arginine. No, it's a specific sequence that's rich in lysine and arginine. There is specificity. Just because you have lysines and arginines does not mean you go to the nucleus, it's specific. The nuclear import receptor will also bind to proteins called nucleoporins in the nuclear pore complex and then mediate the transport to the nucleus. If you look at the figure down here that shows you simple cargo protein, notice it has a nuclear localization signal, specific sequence rich in lysine and arginine. You can see this cargo protein will bind to this nuclear import receptor. Nuclear import receptor recognizes that NLS. Here is a different cargo protein. Notice its NLS, the nuclear localization signal, is different from this one. It can still be rich in lysine and arginine, but it's a different sequence. And because it's a different sequence, it's recognized by a different nuclear import receptor, a blue one versus a green one. Right, so again, you have those signals that say, take me to the nucleus. It's a specific sequence rich in lysine and arginine. But notice it's not going to be the same sequence in all the different proteins, right? This one looks like an arrow. This one over here looks like a blob. You'll be recognized by different nuclear import receptors, a blue one versus a green. It's the same basic concept for nuclear export. You look at nuclear export. In this case, the cargo, the protein that moves, because we're talking about nuclear export, is going to leave the nucleus. It needs a nuclear export signal, an NES. Examples of proteins that have this would be newly assembled ribonucleic ribosomal subunits, RNA proteins with regulated nuclear import and export. The NES, the nuclear export signal, is going to be bound by a nuclear export receptor. And after it binds to it, it'll bind to the nucleoporins, the proteins in the nuclear pore complex to transport it. Okay, so again, here is the protein. This is the cargo protein, the protein that moves. Notice it has a nuclear export signal, specific signal that says, take me out of the nucleus into the cytosol. It will be recognized by these nuclear export receptors. It'll bind to it, and then the nuclear export receptors will bind to those nucleoporins in the nuclear pore complex, take it to the cytosol. Now, the overview of nuclear import and nuclear export is shown on this slide. So what I want to do here is I just want to take you through an overview of these, and then over the next couple of slides you have all the steps. I'll take you through it again, because I really think for this particular part it helps just to take a step back, look at the overall picture, and then over the next couple of slides take lots of notes, and then we'll go through it again. So basically I'm just reading for you what's on the next couple of slides. Now on the left, if you just look up here, this is nuclear import, so going into the nucleus. On the right, this is nuclear export, leaving the nucleus into the cytosol. This side is the cytosol, this side is the nucleus. So nuclear import, you're going from the cytosol to the nucleus. So taking a look at nuclear import, here's the cargo protein. It's a protein with a nuclear localization signal. It says, take me into the nucleus. Here is a nuclear import receptor that recognizes it. So the nuclear import receptor will bind to it, and then it will bind to nucleoporins, proteins along the nuclear pore complex. It'll take the protein inside. Now, you don't just release the cargo on the inside. It's really a displacement. And you'll see this again in a bit. Here is a green protein called RAN, and the green protein called RAN is bound to this red GTP. So RAN protein bound to GTP will essentially displace the cargo. You'll deliver the cargo. Now you have the nuclear import receptor bound to RAN, bound to GTP. This gets taken all the way back to the cytosol, where RAN will cut GTP to GDP and P, and you'll get release. Whole process starts again. So, quick summary. Cargo with the nuclear localization signal binds to the nuclear import receptor. They bind to nucleoporins, move everything inside. Now you don't just release it. RAN protein bound to GTP will displace the cargo, delivering the cargo. Now RAN protein is stuck to the nuclear import receptor. You move it back into the cytosol to recycle it. RAN will cut GTP to GDP and P. Pretty simple. Now you also have nuclear export. So again, you have all these notes on the next couple of slides. Now for nuclear export, what you actually see is the nuclear export receptor binding to the cargo and RAN protein bound to GTP. They all bind at the same time in the nucleus. Everything gets moved out into the cytosol. Now, at the cytosolic side, RAN will cut GTP to GDP and P, and you break everything up. Cargo gets delivered, nuclear export receptor gets released, this thing gets decided. So again, everything binds on the cytosolic side, or sorry, on the nucleus side. You move it to the cytosol, RAN will cut GTP to GDP and P, everything gets released. Now, if you compare nuclear import and nuclear export, they both have something in common. On the nuclear side, you need RAN protein bound to GTP. So right here, you need RAN protein bound to GTP to release the cargo. Right here, you need RAN protein bound to GTP to bind to everything. And then when you move to the cytosolic side, you need RAN to cut GTP to GDP and P. And same thing over here, you need RAN to cut GTP to GDP and P. Both are consistent. Both need RAN bound to GTP on the nuclear side, and then both will cut the GTP to GDP and P on the cytosolic side. So that's where we're going to start, and then you're going to have all the steps I just described. Okay, so RAN is the first thing we're talking about. RAN cycles between GDP bound and GTP bound. And for RAN to cut GTP to GDP, you actually need a bit of help. again, the first thing, before we talk about all the steps I just showed you, the first thing we want to talk about is a specific protein called RAN. Now you can call RAN by a couple of different names. You can say RAN. You can also say RAN protein, right? It's a protein called RAN. You can also call it RAN GTPase. Same thing. So RAN GTPase is also RAN. It's also RAN protein. Now RAN GTPase, also known as RAN or RAN protein, it's what we call a GTPase. It's supposed to cut GTP into GDP. That's what GTPases do. Cut GTP, triphosphate, into GDP, guanosine diphosphate. However, RAN, also known as RAN GTPase, even though it's supposed to do that, it is horrible at its job. It needs help. So in order for it to do its job, in order for it to cut GTP to GDP, it needs help by something we call a RAN gap. That's a second protein. And RAN gap stands for RAN GTPase activating protein. So it's going to activate the protein we call RAN, or activate the protein we call RAN GTPase. It says, do your job. It will stimulate GTP hydrolysis by RAN. Now, if RAN is bound to GDP, you actually can recycle it. So you can use what's called a RAN-GAF, a guanine nucleotide exchange factor, to help RAN, also known as RAN protein, also known as RAN GTPase, release GDP for GTP. What you'll see is the RAN-GAP proteins in the cytosol and RAN-GAF is in the nucleus. And what you actually do is that you'll maintain a very high concentration of RAN GTP in the nucleus, low concentration of RAN GTP in the cytosol, and you need that for the direction of transport. Okay, so it looks like a lot of text, most of it confusing, but you'll see it become quite clear on this figure. So if you look at this figure, this side is the cytosol, this side is the nucleus. Here in green is RAN protein, also called RAN GTPase, also just called RAN. And right now, RAN is bound to GTP. And we said this RAN GTPase, it's supposed to cut this GTP into GDP, but it's horrible at its job. It doesn't really do a good job. So it just kind of stays with GTP. Now, whether you have nuclear import or nuclear export, doesn't matter which one, eventually what happens is RAN bound to GTP will move into the cytosol. With either nuclear import or nuclear export, that's what happens. Now, in the cytosol, you have lots of RAN-GAP. And we said RAN-GAP is the GTPase activating protein. It's the one that says, RAN, do your job. So it's going to make RAN cut GTP into GDP and P. Eventually, what happens is RAN bound to GDP will move back into the nucleus, where in the nucleus you have a RAN-GAF, which helps RAN exchange GDP for GTP. So it's an exchange, guanine nucleotide exchange factor, where again you have RAN bound to GTP. That is the standard RAN cycle. You need lots of RAN bound to GTP in the nucleus, less of it in the cytosol. Okay, so very quick review, because you'll see how this comes into play with nuclear import and export. RAN protein, also known as RAN GTPase, also known as RAN, is bound to GTP. Whether you have nuclear import or export, it gets moved out into the cytosol. At the cytosolic side, there's a RAN gap that makes RAN do its job. So RAN will cut GTP into GDP and P. RAN bound to GDP will eventually come back into the nucleus, where a guanine nucleotide exchange factor, RAN-GAF, will help it exchange GDP for GTP. Cycle continues. Again, RAN bound to GTP, right? RAN protein bound to GTP, is going to go to the cytosol with the nuclear import and export receptors. So step one, that is this thing right here, right? So step one right here. Whether nuclear import or nuclear export, RAN protein bound to GTP goes to the cytosol. And then RAN protein bound to GDP will go to the nucleus. Step two right here, that's this one right here. NTF2 is the special protein that takes RAN bound to GDP back into the nucleus. Okay, so with this in mind, I know you might not have this completely down, but with this in mind, you're going to see the next couple of steps and you'll see how it all fits together. So these are the steps. This is half of the slide that I showed you initially. These are the steps for nuclear import of cargo. So just step by step, you can go through. I'm going to show you on the figure the step-by-step process, and you have all the steps here, right? So I'm just going to basically read them to you and show them to you. So again, this side's the cytosol, this side's the nucleus. You're talking about nuclear import, so you're moving cargo into the nucleus. So step number 1 says the nuclear import receptor binds to the cargo in the cytosol. That's right here, right? Here's the cargo, here's the nuclear import receptor. They bind to each other. Step number 2, right here. Receptor plus the cargo move into the nucleus, right? Into the nucleus. Step number 3, RAN protein bound to GTP. So RAN and GTP binding causes the cargo release. Step 3 is right here. Step 4, the MT import receptor plus RAN bound to GTP move to the cytosol. So this is step 4. And then step 5, RAN binding protein and RAN gap promote GTP hydrolysis and the release of the nuclear import receptor. Now notice in this step, on the figure, they actually don't show you the RAN gap. So on this side, remember, there's a RAN gap, GTPase activating protein that makes RAN do its job. There's also another protein called RAN binding protein that you need to make RAN do its job. You actually need 2 proteins. The RAN GTPase activating protein and a RAN binding protein, both on the cytosolic side here, to make RAN cut GTP to GDP. So those are the steps. Now I'll show you again one more time, but this is in the animated form. And this is actually a flash animation that we'll post on the web, and I would highly encourage you to look at this. It's going to be in the lecture section on Portal. And I'll show it to you step by step. It really shows you everything I told you step by step. So this side's the cytosol side, this side's the nucleus side. Again, you can choose nuclear import or export at the bottom, so we're choosing nuclear import because that's what we're talking about. This, the legend is on the side here, this is your cargo protein, the protein that moves. Here is the nuclear localization signal, and here's the nuclear import receptor. So let me just play it. So the nuclear import receptor is going to bind to the cargo protein. Then you'll move it into the nucleus. Then RAN protein bound to GTP will displace it. And then you have a RAN gap and RAN binding protein to bind to it to cause RAN to do its job, to cut GTP to GDP. Now NTF2 is going to take RAN bound to GDP back into the nucleus. And in the nucleus, you have a RAN gap to help RAN do the exchange, GDP for GTP. And the whole cycle continues, right? So step by step, they show you everything here. All right, now you have the same kind of figure for nuclear export of cargo. Again, all the steps on the left here. So we're talking about nuclear export, so you're going from the nucleus to the cytosol. This side's going to be the nucleus down here. Top side is going to be the cytosolic side. Step number 1 is right here. It says nuclear export receptor binds to RAN bound to GTP and the cargo in the nucleus. Step number 2 over here, receptor plus cargo plus RAN GTP move to the cytosol. Step number 3 is right here. RAN binding protein and RAN gap promote GTP hydrolysis. So the RAN binding protein, RAN gap, going to make RAN cut GTP to GDP and P. You'll get the release of the cargo. You'll get the release of the nuclear export receptor. And the nuclear export receptor, step number 4, can go back into the nucleus. Whole process gets recycled. Again, on this slide, they do not show you the RAN binding protein and the RAN gap, but again, they are very necessary there on the cytosol side. So again, showing to you in animation form, we have the same flash animation. So when you find this online on the lecture section, just go to the bottom, choose nuclear export. Again, cytosol side, nucleus side, here is the cargo with the nuclear export signal. You have the nuclear export receptor shown for you as an S form. So first they're going to bind to one another with RAN bound to GTP. Move along the nuclear Bohr complex. Then RAN binding protein, RAN gap, will make RAN do its job. It's going to make RAN cut GTP to GDP. You can see everything gets released. And then NTF2 is going to take RAN bound to GDP back into the nucleus. And then in the nucleus, a RAN gap will help RAN exchange to GDP for GTP. Whole cycle continues again. Alright, so that's essentially nuclear import and nuclear export. Lots of steps in the cycle, but make sure you go through the animation, go through the steps one at a time, and you'll be okay. Couple of quick, quicker questions. So first, where is RAN bound to GTP predominantly found in the nucleus or in the cytosol? If you can. Okay, nice, yeah. So RAN bound to GTP is predominantly found in the nucleus. Question number 2. What is the function of nuclear transport factor 2, NTF2? To transport A, cargo to the cytosol, B, RAN and GDP to the nucleus, C, cargo to the nucleus, D, NTF to the cytosol. So buzz in if you can. Okay, nice. And B. And only one person chose this stuff. So B, again, RAN GDP to the nucleus, right? So NTF2 takes it back. Now, be a little careful here. This is NTF2. NTF2. It's used to transport RAN GDP to the nucleus. NTF2 is not the same as NFAT. This is something totally different. So do not mix those up on the exam. It's actually quite easy to mix those two up. Now, for this slide, you can see a cycle here going from the cytosol to the nucleus and back. Kind of what makes this slide a little bit more easy to understand is if you kind of divide it up like this. So it is a cycle, don't get me wrong, but it's a little bit more easy to understand if you take a look at it, divide it as I show you here. As we'll see, NFAT, as in nuclear factor activated T cells, is actually a transcription factor. A transcription factor. And it actually has both nuclear import and nuclear export signals. It's a transcription factor, and it has both nuclear import and nuclear export signals. Alright, so let's see what happens in a resting T cell. So imagine a T cell here is resting as it has not encountered an antigen, right? Doesn't see a bacteria. This T cell is not going to be activated. It's just going to be doing its thing. Not activated, no bacteria, no antigen. Eventually what happens is it will lower its calcium concentrations. We won't talk about how it does, it's quite complicated, but you get a lowering of calcium concentrations. What happens is an active protein kinase will use ATP and phosphorylate NFAT. Now if you use ATP, use the protein kinase, and you phosphorylate NFAT, you're going to change its structure so that it exposes a nuclear export signal. When you phosphorylate, when you change the structure, when you expose a nuclear export signal, This whole thing will be exported from the nucleus to the cytosol, using exactly the same mechanisms we just talked about. So there's going to be RAN down to GTP, nuclear export, receptor, etc. But the whole thing gets moved out. Now, if this thing is in the cytosol, it will not activate transcription. Right? It's a transcription factor, should bind to DNA, but if you're in the cytosol, you're not going to activate transcription. So again, resting T cell. T cell is not activated, there's low calcium, protein kinase is going to take ATP, you're going to phosphorylate this protein, changing its structure, you will expose a nuclear export signal. Using all the mechanisms we just talked about, you move this from the nucleus to the cytosol, where it's not going to be active, right? Transcription factor in the cytosol, there's no DNA there, you're not going to activate transcription. Now that's very different when the T cell encounters an antigen, maybe it encounters a bacteria, it's going to be activated, it wants to attack. So when it's activated, it will elevate its calcium concentrations. It will activate a protein called calcineurin. And calcineurin is a protein phosphatase, meaning it removes phosphate groups. And if you remove the phosphate groups, you will change the structure of the protein. It will expose a nuclear import signal. And using all the mechanisms we just talked about, you will import this into the nucleus, where you can activate gene transcription. So again, activated T cell, you're going to get high calcium, which will mean activating calcineurin, which is a protein phosphatase, so it will remove phosphate groups. It'll change the structure of the protein to expose a nuclear import signal, bring this whole thing into the nucleus, using all the mechanisms we just talked about. This will now activate gene transcription. So you can kind of get the picture, hopefully, with NFAT, with this one protein, has both nuclear import signals and nuclear export signals, but depending on phosphorylation, different ones are exposed. And for other proteins, you can modify them through different ways, and you get different signals exposed as well. So again, NFAT with high intracellular calcium, you'll get nuclear import. Low intracellular calcium, you get nuclear export. Now, what they do in the experiment shown for you on the bottom of the slide in the video is that they take NFAT and fuse it to GFP. You add a calcium ionophore, so basically a calcium ionophore helps calcium pass through the membrane. So if you add a calcium ionophore to get calcium to pass through the membrane, there's going to be more calcium inside, then you can monitor the fluorescence. And that's exactly what they do in this picture here.
Nuclear import and export can be directly visualized in living cells that express the green fluorescent protein GFP fused to the gene regulatory protein NFAT. NFAT is normally localized in the cytosol and excluded from the nucleus. But when the cytosolic calcium concentration is raised, NFAT migrates to the nucleus. This is done here experimentally by adding an ionophore that allows calcium to enter the cells from the medium. Upon removal of the ionophore, calcium levels return to normal, and NFAT is exported from the nucleus. Re-addition of the ionophore triggers re-import of NFAT.
Okay, so we can actually see the movements. The other thing we want to talk about, so this is that was gated transport, going back and forth between the nucleus and the cytosol. The next thing will be transmembrane transport, and that's going in one direction from the cytosol to the ER, mitochondria, plastids, peroxisomes. Here you're going to need protein translocators. Proteins usually, not always, but it's usually unfolded. Protein translocators will help transport the protein across the membrane. Proteins usually unfold, not always. Mitochondria and chloroplasts have their own genome and ribosomes, but in fact most of the proteins they have are nuclear encoded. We get translation in the cytosol, import into the organelle. And for mitochondria and chloroplasts, it's post-translational sorting with unfolded proteins. Some of you will remember that mitochondria and chloroplasts do have their own genome, but obviously obviously their genome's not that large, so a lot of your nuclear DNA does encode for proteins that go to the mitochondrial chloroplast. Protein sorting in that case is always post-translational, so it's completely translated. Proteins will remain unfolded in the cytosol by association with HSP70 chaperones. HSP70 chaperones, these are the ones we learned about last class. This is not the one that looks like two barrels or two buckets. It's not the isolation chamber one. It's actually the other one. Now remember, there's timing to these things. So HSP70 proteins, they help other proteins fold, but there's timing, as in they keep it unfolded until the right time and then release the right amino acid. So you can think of it in this case as binding to them, keeping it unfolded till the right time. There's time. Now here is your standard mitochondrial layout. Just to remind you guys of your mitochondria, you have your outer membrane, that's a phospholipid bilayer. You have an inner membrane, that's another phospholipid bilayer. You have an intermembrane space, the space between the outer and inner membranes, and then the matrix space in the middle. That's similar, but not quite exactly the same as chloroplasts. Chloroplasts have an outer membrane, that's a phospholipid bilayer. You also have an inner membrane, that's a phospholipid bilayer. You also have an intermembrane space. The stroma, or the matrix space, is in the middle, but now you have a third set of phospholipid membranes. It's a thylakoid membrane. It's another phospholipid bilayer, and inside you have a thylakoid space. So keep that in mind for chloroplasts versus mitochondria. First thing we want to talk about is sorting to the mitochondria. Look at your figure. This side is the cytosol. So you have an outer mitochondrial membrane right here. And over here you have an inner mitochondrial membrane. Here is the protein. Again, it's going to be post-translational sorting. This protein will be unfolded. They don't show you the HSP70s, but they're there. There's a specific signal sequence that takes this protein that says, take me to the mitochondria. You'll take the protein to the mitochondria to what we call these TOM complexes. So if you look at here, TOM complex, that's a protein translocator. I'll tell you what TOM stands for because it's going to help you remember it. TOM is translocase of the outer membrane. TOM is translocase of the outer membrane. So if TOM is translocase of the outer membrane, then TIM is translocase of the inner membrane. And that will tell you exactly where they are. Translocase of the outer membrane versus translocase of the inner membrane. A precursor protein, the signal for taking you to the mitochondria is a specific N-terminal amphipathic alpha helix. There is specificity. It's not just any N-terminal amphipathic helix. Okay, so basically what they're saying down here is, well, you have your precursor protein as a signal sequence. Again, that's an N-terminal amphipathic alpha helix, but it's not just any N-terminal amphipathic helix. There's specificity, specific kind, specific sequence. That will bind to a receptor in the TOM complex, right, translocase of the outer membrane. The TOM complex, translocase of the outer membrane, will transfer the protein to the TIM complex, the translocase of the inner membrane. When you're in the matrix space, then a signal peptidase will cleave off that signal, right? This protein now is in the matrix. You no longer need that signal. You're already taken to the mitochondria. So this is actually quite interesting. You'll notice this is the N-terminal portion that you cut off. So sometimes students will ask, well, is the N-terminus of all proteins, is it always methionine, right? Methionine start codon, is it always methionine in the N-terminus? Well, maybe it used to be. Like for this, you used to have a methionine. But after you cut it off, you might not have the methionine at the N-terminus anymore. You might not necessarily have that same amino acid. Now, proteins can obviously be further sorted from here, but we won't go through the further sorting mechanisms. This is kind of the general mechanism. Now, that's the mitochondria. Chloroplasts are actually very, very similar. N-terminal amphipathic alpha helix. signal can get cleaved. You can have a separate phthalate-coid signal sequence. So if you look at kind of the first thing you wrote out here, for sorting to the chloroplast, the proteins have an N-terminal amphipathic alpha helix. That has to be different from the mitochondrial signal sequence, right? Mitochondria, to sort to mitochondria, it's also an N-terminal amphipathic alpha helix. To sort to chloroplast is an N-terminal amphipathic alpha helix. But there's specificity in the sequence. It's not the same. Like if you think of plants, for example, plants have both mitochondria and chloroplasts. Mitochondria, that's for ATP generation. Chloroplast, that's for photosynthesis. You're not going to have the same proteins at both mitochondria and chloroplasts. So obviously, if they both use N-terminal amphipathic alpha helices, there's a specific sequence to take you to mitochondria. There's other specific sequences to take you to chloroplasts, right? So even though it's N-terminal amphipathic alpha helix, they're different sequences for mitochondria, not the same thing. Now, here's your standard chloroplast setup. This is the cytosol side. This is the stroma side. You have a chloroplast outer membrane. You have a chloroplast inner membrane. You as well have the phthalate-coid membrane. Here is your precursor protein. This protein, again, is going to be post-translationally sorted. You have those HSPs there to keep it unfolded. Notice you have that red chloroplast signal sequence, that N-terminal amphipathic alpha helix. As well, in this case, you have this yellow phthalate-coid sequence. You don't need that phthalate-coid sequence, of course, unless you're going to the phthalate-coid. Now, you will bind to a translocator in the outer membrane. You will then transfer the protein to a translocator in the inner membrane. Now you are in the stroma. In the stroma, a signal sequence will be cut off. So signal peptidase will cut off the red sequence. You're already in the chloroplast. You don't need it anymore. If you cut off the red sequence, you are now exposing the phthalate-coid sequence. And in your textbook, there's actually, they'll show you 4 different ways by which you can move this protein into the phthalate-coid. You don't need to memorize the 4 different ways, but you can read about it if you're so interested. But if you have that sequence, you can then move it into the phthalate-coid, where you then cleave the phthalate-coid sequence. So again, whole thing gets made, associated with the outer membrane translocators, move it to the inner membrane translocators, cut off the signal sequence, and if you're the type of protein that goes to the phthalate-coid, well then you'll have the exposed phthalate-coid sequence. That will make it go into the phthalate-coid, and then you'll cut off that signal sequence. So that's the chloroplast. Last thing is the peroxisome, and this is fairly easy to understand. It's a C-terminal SKL sequence. Proteins in this case are usually clear. So you can see this is your standard protein. Here's the cytosolic side. Protein's going to be folded in this case. This protein, because it has the C-terminal SKL sequence, is going to be taken to the peroxisome. So you have serine, lysine, leucine here. That's going to bind to a soluble receptor. Soluble receptor will take it to the peroxisome, to this docking protein. You'll then transfer the protein to a translocator. Translocator will translocate this thing. You're going to folded protein with SKL at the C-terminus, binds to the receptor. The receptor will take it to the docking protein. Then you transfer it to the translocator, which transports it in. Then you have the protein. All right. Now, you can go take a look at this animation online. I'm not going to show it to you here, just because I didn't find it that helpful, but you should definitely check it out just to make sure if it course, for you. What I actually did find very helpful was this review table. So notice, so I'm going to let you guys fill this out, but take a look here. Destination from cytosol to the nucleus, to the mitochondria, to peroxisomes. Don't forget, we also have chloroplasts. That's not given to you. We also have chloroplasts. The mechanism to the nucleus is gated. Signal is NLS, nuclear localization signal. Protein is folded before the transport. So I'm going to leave this up here for you to copy down. Don't forget to fill in the chloroplasts. That's not a row here, but you should know chloroplasts as well. And we're going to take a 10-minute break from here. If you're done copying, just be nice to your neighbor. Don't knock them over while writing and spill their drink. Okay, so 10-minute break. I have 7:10. We're resuming at 7:20. Okay, 7. Questions or what?